Today, on the BMC podcast, we have been talking to Dr. Haig Kazazian, Professor of Pediatrics and Molecular Biology and Genetics at the Johns Hopkins University School of Medicine. Dr. Kazazian is a pioneer in the field of genetics, identifying and investigating the biology of line 1 retrotransposons in human beings and mice. He serves as an editor-in-chief on the BMC journal Mobile DNA. He was also named as one of the first plaintiffs in the Supreme Court's 2013 ruling that companies cannot patent parts of naturally occurring human genes. This was just one point, however, on a long and storied career. We spoke to him about his professional history and got a whistle-stop tour through the world of transposable elements in DNA and their relation to disease. Thank you very much for joining us today. To start off, could you give us a brief introduction, a bit about your research background and training, and how you became interested in transposable elements in DNA?
Well, I became interested in a somewhat serendipitous way, bumped into them in our work. But I can tell you that I was from Toledo, Ohio. I went to public schools in Toledo and then attended Dartmouth College, where I was pre-med, and started at Dartmouth Medical School after my junior year. That's what almost all the Dartmouth fellows did. And then I transferred. I had to transfer. It was a 2-year school to Hopkins, where I finished medical school. Then I was very interested in pediatrics, so I took a pediatric internship and residency at the University of Minnesota, and but after the first year of that house staff training, I decided that I wanted to go into genetics, and this was sparked by a seminar course I had taken with Barton Childs, the father of pediatric genetics, at Hopkins in 1961, actually. And I wrote to Childs and asked him where I should take a fellowship in genetics, and he gave me a few possibilities, but he also mentioned Hopkins, so it turned out I decided to come back to Hopkins and work with Childs for my fellowship after 2 years of house staff training. And then after another year and a half of that, I was about to be drafted into the Army, and I found a position at NIH with Harvey Atano, a major player in the hemoglobin field at the time. And I trained with Atano for another 2 years down there, and then came back and did a third year of residency at Hopkins before I joined the faculty at Hopkins in pediatrics. And I worked on the hemoglobin system, regulation of the hemoglobin chains for a number of years. Had very good luck solving the mutations in the thalassemia syndromes, especially with a huge amount of help from Stuart Orkin at Harvard and Stylianos Antonarakis, who was a postdoctoral fellow with me in the early 80s. But we had pretty much solved the mutations in the beta globin chain of hemoglobin, and that was about 84 or so, and we decided to start looking in a bigger gene where there wasn't selection for the particular mutations as there was in thalassemia in the malarial regions of the world, and so we got a hold of the very large factor VIII gene, which is mutated in hemophilia A. And in the mid 80s, we started to analyze the mutations in the factor VIII gene. And that's how I happened to get into transposable elements, because we found an insertion of a line 1 element in 2 patients out of 240 that we were analyzing for mutations in factor VIII. It was very serendipitous, as I've said before, and we were quite lucky. But we were also lucky that just in the lab above us, Alan Scott was pulling out line 1 elements from the human genome that were fixed in the genome, and he was sequencing those elements, and he had made a consensus sequence of the line 1 element, the full-length element, which was a little over 6 kilobases in size. He had done this after sequencing some 20 or so different human elements. And with this consensus sequence, that was extremely helpful, because once we found, well, actually, to go back, we were lucky enough to find that the insertion that we had was a line element, because Alan had probes for line 1 and ALU, and we could just go to him and get those probes, and then detect the fact that our insertion was a line 1 using his line 1 probe. Then we decided to try to isolate the precursor of the largest line 1 insertion that we had, which was just under 4 kilobases. And I was fortunate enough to look at Alan's consensus sequence and find a region that we could analyze with an oligonucleotide that was different from the sequence of the insertion. I'm sorry, it was different from the consensus sequence was different by 3 nucleotides out of 20 nucleotides from the insertion. So we thought if we made a probe for the insertion, that it would pick up very few elements in the genome, because the consensus sequence was different. So we did that, and indeed, instead of seeing a smear with the probe, we actually found specific bands, 6 or 7 bands. And when we looked at the 2 parents and the youngster with hemophilia who had the insertion, we found that youngster had a band which was not seen in the parents, and that was the band of the insertion. And that got us onto the possibility, because we had specific bands, of isolating the precursor to the insertion, which we were able to do. I'm sorry, that was a little long-winded.
That's quite all right. I think it's a bit of a detective story then, as you kind of tracked this down. Following on from this kind of initial discovery of the transposable elements and what they might mean, how did you come to the realization that there was a connection between these and human disease?
Well, that was right off the bat with finding the insertion. That insertion, I should say, was in an exon of the factor VIII gene, and it was disrupting the production of normal factor VIII. The child that had that insertion had no factor VIII activity and had hemophilia. And that was the same for the second insertion that we had at that time. The insertion was in this large exon, and it disrupted activity. Actually, there was a third insertion, but it was in an intron, and we were able to show by tracking the inheritance of that insertion that it was not related to the hemophilia in the child. After that, though, we at the time we were running a prenatal diagnostic clinic, which was doing DNA analysis, and we were doing DNA analysis for other diseases, and one of those diseases was Duchenne muscular dystrophy, and that gene is enormous. That compared to the factor VIII gene, which is around 200 kilobases in size, the Duchenne gene is 2 megabases. And in analyzing mutations in the Duchenne gene, we found another mutation that was in an exon that disrupted the Duchenne gene, and that led to an interesting finding that that particular insertion had carried along with it sequences in its back end, its 3' flank, and so that led to the possibility that not only the L1 was retrotransposed or jumped, but it could jump with other sequences along with it. And it now turns out that from those first 2 or 3 insertions causing disease, there are, if we count the ALU insertions along with the line 1 insertions and insertions from a primate-specific element called SVA, that there are now some 150 or more insertions known that cause disease.
Okay. Almost mechanically speaking, how exactly is it that these insertions are causing the disease? What are they doing in the cells, say, for example, in hemophilia, that is causing this disease?
Well, they go. If they go into an exon, they're putting in an extra 1,000 up to 4,000 or 6,000 new base pairs, and they change the reading frame for translation of the protein. So you end up with no good protein made. There are other mechanisms that have been found too. They may alter splicing. They may go into an intron in the gene and then produce new splice sites within the transposable element. New splice sites in ALUs or new splice sites in SVAs or line 1 elements. And there are actually a bunch of different mechanisms, and there's some we don't really understand, where the insertion has gone into say a 5' exon, a 5' intron of the gene, and we don't really understand why it is turning off the messenger RNA production in that gene.
So aside from the ones you've cited before, what are the other major diseases that have been identified that can be attributed to these transposable elements?
Well, you have to understand that these are single cases of the disease. Each insertion is a single case, and I said there's something like 150 cases known, but there are a few. There's a couple of diseases where essentially the whole disease, all the cases of the disease, are due to a transposable element insertion. One of the good examples of that is a muscular dystrophy I guess it's called Fukuyama muscular dystrophy. It's found in the Japanese, and it's found in maybe 90% of the disease is in the Japanese, and there is one mutation. It's an SVA insertion into a very 3' sequence of the gene that causes an unusual splicing event and knocks out the gene. This is an X-linked disorder, just like hemophilia A, and so it only takes one hit in the child, in the male, to produce the disease. And it turns out that this particular SVA insertion has been in the population, the Japanese population, for a long time. So nearly all the cases of the disease in Japan, this muscular dystrophy, are caused by this SVA insertion. And people now are working the lab in Japan is working to figure out a good treatment which blocks the splicing into the SVA.
So that leads me nicely on to the sort of final point then. With the identification of these cases, what are the mechanisms for diagnosing and treating these diseases now we know the root cause?
Yeah, so as I said, there is this development of a treatment for this particular muscular dystrophy in the Japanese. But interestingly, there was a very interesting report in a single case of Batten's disease, which is a very bad neurodegenerative disorder, where there was an SVA insertion again, which caused aberrant splicing, and the investigators at Boston Children's Hospital developed a specific oligonucleotide treatment to block that splicing, and this was reported back in October, and I believe that child has shown considerable improvement with that new treatment. And yet this is a treatment for a single case. So there's no other cases known with this particular mutation. And so this SVA has just arisen very recently in time. And treatment for this single case developed and looks like it's somewhat successful. I don't know how the child is doing neurologically at this time, but they reported it, and it gives hope for this kind of situation.
Is the outlook for these kinds of treatments then going to be more these personalized therapies, or would there be a way to scale it out?
They may well be, yes. Yes, as I said, there are very few of these conditions where there is more than the one case in a family. There's another condition that I can tell you about that has pretty much all the cases due to the same retrotransposition event. But almost all the remaining 150 or so cases are single cases where the insertion is found just in the one patient, and so that may take a more personalized approach. And at this point, I think that for instance, the case that I was telling you about, the Batten's case, that took about a year or so to develop the treatment and to try it in a single case.
Okay, wow.
Interesting, huh?
Yeah, it feels like every treatment's going to need its own lab of professionals and experts
Yeah.
working on it. How big is the field of people working on it? Is it something that's growing because of the need to identify and treat more of these?
Yes, it seems that it has been growing. When I started work in the field, there were maybe less than a handful of labs that were involved in it, and I had to get help to learn about the field from Maxine Singer at the time. That was in the late 1980s, and now I would say that there could be 100 or more labs working in the field, especially if we think about non-human. I'm talking about labs working in transposable elements in general, plants, bacteria, lower eukaryotic organisms, and human. There's got to be at least 100 labs, I would think.
Oh, excellent. If there are any other sort of closing points or things that you think would be interesting to the kind of wider scientific audience.
Well, I think it's a fascinating field to think about pieces of DNA in essentially every genome that are moving around in the genome. And as far as the human or mammalian genomes go, about half of the genome is dead remnants of transposable elements. I'd say, when I first went into the field of genetics, we thought at that time, back in the 60s, that the whole genome was protein-coding genes. And now we know that the protein coding part of the genome is about 1.5% of the genome. We thought it was 100%. And we had no idea that half of the genome was going to be remnants of transposable elements. And it turns out that only a very, very small minority of those transposable elements are actually active in the human genome. There's maybe in the average human, there's perhaps 100 to 150 line 1 elements that are active, and small numbers of ALUs and SVAs that can be jumped by line 1 elements. So the genome is a different place from what we thought it was.
What's
the
benefit of them if we have this 100 or 200 that are actually doing it? What's the evolutionary purpose there?
We don't know. I mean, we don't know that there's any real benefit. Obviously, they provide, from an evolutionary point of view, sequences that can be used for other things, exapted sequences. But we don't know that they have an actual function in the individual. There have been a few reports in the last couple of years of perhaps they have a role in early development or in other parts of our physiology, but it's really not clear. I think the evidence is still out, and we'll just have to see. But I think many investigators believe that they don't really have an actual function. So it's hard to know why we're living with all these elements that can jump around, and
that
all the organisms are.
Well, okay then. Thank you very much for your time, Haig. That was a fascinating tour through the
You're welcome.
The history of these.
My pleasure, Alex. Good. My pleasure.